Greetings to my fellow examiner. My name is Benny Evan and I'm going to present my final year project titled Comparative 3D Predicted Protein Structures and Functional Analysis of M.sagu kinase 2. Abstract. Enzyme kinase widely distributed in nature as they are found in various species from all of the kingdoms. The enzyme kinase break down the chitin. In the study, 3D predicted protein structure of M.sagu kinase 2 was used to compare with main kingdoms of organisms which are plantae, animalia, fungi, and bacteria. In this study, bioinformatic tools were used. Introduction. The aim of this report is to conduct analysis of 3D predicted protein structure and functional analysis with M.sagu kinase 2 with kingdoms of Animalia, Plantae, Fungi, and Bacteria. The objectives are comparing the protein structures, identify the domains and motifs, and identify the functions of each protein towards each organism. Literature review. Chitinase are enzyme that is responsible for degrading chitin, and there are two main groups of chitinase which are endochitinase and exochitinase. There are two families of glycosyl hydrolases which are families of 18 and family of 19. In this study, M.sagu kinase 2 was used to compare with other kingdom of chitinases. Protein. The protein function will depend on the three-dimensional structure of the protein. Protein motifs can be defined as short conserved sequence pattern related. Meanwhile, protein domain can be defined as autonomously folding functional of a protein. Enzyme. Enzyme is an important component in biological reaction as it speeds up the reaction. The enzyme works by catalyzing the chemical reaction, then converting the substrate to a product. Enzyme chitinase are grouped under glycosyl hydrolases, and chitinase are hydrolytic enzymes that break down the glycosidic bond in chitin. Bioinformatic tools is a technique that is significant in protein analysis. Example of tools are NCBI. NCBI, one of the main leading sources that provide public biomedical database and other information such as protein sequence. Next is BLAST. BLAST analysis, the function of it is to search for regions of local similarities between sequences such as amino acid sequence and nucleotide sequences. Another example of bioinformatic tools are UniProt KB, Protein Data Bank, Multiple Sequence Alignment which is COSL-W, FIRE2 Swiss model, and Chimera. Materials and methods. For the data, amino acid sequence of the M.sagu kinase 2 was retrieved from NCBI. A BLAST analysis is performed to find the local similarities among the amino acid sequence across the kingdom, where M.sagu kinase was used as a query sequence. From the BLAST analysis, two plant species were chosen that have higher similarities to M.sagu kinase 2 amino acid sequence. These were repeated with other organisms which were Animalia and fungi. The rest of the samples from each kingdom are chosen from UniProt database. For the sample from kingdom bacteria, all 5 samples are chosen from UniProt database. For the 3D predicted protein structure of M.sagu kinase 2 and other kingdom, FIRE2 web server and Swiss model web server was used. For the FIRE2, the template model or model homology which had high confidence and high coverage were chosen. For multiple sequence alignment, across kingdom with M.sagu kinase 2 were performed. The multiple sequence alignments are performed to find the similarities and identity of percentage of amino acid sequence across the kingdom. Comparisons of 3D predicted structure of protein was performed by using Chimera. The comparison will be matched from each 3D structure of the kingdoms and superimpose the structure with 3D structure of M.sagu kinase 2. Result and discussion. Based on the discussion of sequence analysis of M.sagu catenase 2 using Interpro, the M.sagu belongs to the catalytic domain of glycoside hydrolase family of 18. Biological process of M.sagu catenase 2 is carbohydrate metabolic process. The domain type of M.sagu catenase 2 is GH family 18 or glycoside hydrolase family 18. Catenase 2 is found in mammals, plant, viruses, bacteria, and fungi which belong to GH family 18. Meanwhile, plants found in GH family 19. In sequence analysis of kingdom animals, plants, fungi, and bacteria, almost all of the organisms and M.sagu catenase 2 belong to the catalytic domain of glycoside hydrolase family of 18. The GH18 family catenases are distributed across the organism, and some of the kingdom that does not have chitin such as plants and mammals are included. The roles of catenase enzyme in plant GH19 family is for defensive purposes. Meanwhile, GH18 catalytic domain have various functions such as in kingdom Animalia, insect anatomy is encrusted with a tough layer of chitin that produce chitinases during stage of development. Apart from that, rat and mouse have acidic mammalian chitinase that is associated with the immune system diseases. Next is multiple sequence alignment. Console W was used to align all of the amino acid sequence, and the query sequence is M.sagu catenase 2. Upon analysis, kingdom Animalia have the highest percentage of identity among all of the kingdom. M.sagu catenase 2 and kingdom Animalia have 4.9% identity, which a direct match in the alignment. Next is based on the result for the 3D predicted structures of protein and sequence analysis. M.sagu catenase 2, which was obtained from Fire2 web server, shows similarities with organism which is fern. The confidence level is 100% and coverage is at 87% to the similarities to the M.sagu catenase 2 amino acid sequence. The GH18 catalytic domains have 4 highly conserved regions or signature motifs amongst the amino acids, and GH18 catenase type was first observed in bacteria. Sometimes they are named bacteria type chitinase. Next is based on the result from Chimera software, the superimpose of M.sagu catenase 2 and African oil palm have the smallest RMSD value compared to the other plant species. Root mean square deviation can be defined as 2 corresponding protein structure to measure the similarity between 2 structures of the protein. Therefore, the value of RMSD for both of the 2 structures is 0.001, and if the values is 0, meaning the structures were identical, large value of RMSD means they are not similar. They're not similar. In conclusion, the 3D predicted protein structure of M.sagu catenase 2 has similar in kingdom Plantae, specifically in African oil palm. The RMSD value and the superimpose of the structure obtained from Chimera shows the lowest value, which both of the structures belong to the group GH18 family based on the domain type. Therefore, the existence of bioinformatic tools allow user to make an analysis of 3D predicted protein structure. This is the list of reference, and that's all from me. Thank you.